Hi everyone, so today's video is just going to go over the mock USABO test. So let's see how we did. So the first question asks about basically what modifications to an amino acid in outer membrane proteins would most likely confer resistance to an antibiotic. So in this case, we want to find the amino acid modification that would create the biggest change. And with these problems, we're looking for a change in properties. So like a nonpolar amino acid to a polar amino acid, an acidic amino acid to a basic amino acid, something like that. So let's just look at all of them. So first is lysine to histidine. So this one is from a basic amino acid to another basic amino acid, so it wouldn't be so big of a difference. Glutamine to serine, those are both polar. Now, aspartate to glycine, that is an acidic amino acid to a nonpolar amino acid, so that's a pretty big change. And proline to alanine, nonpolar to nonpolar, not a big change. And then tyrosine to cysteine is polar to polar, so not a big change. So the answer is aspartate to glycine. So for the next question, which of the following do not exhibit quaternary structure? So we're basically looking for proteins that, only have one subunit. So myoglobin, only one subunit. Collagen actually has three subunits, like three kind of fibers that wind together. So it does have quaternary structure. The gamma subunit of fetal hemoglobin, so that's just one subunit, so technically no quaternary structure there. Insulin is made of multiple chains, and tubulin has tubulin is a dimer of alpha tubulin and beta tubulin. So yeah. Next is which is false about fats. So basically all of these are true. Hydrogenation is a method that can be used to artificially saturate fats. You basically bombard it with hydrogens. Unsaturated fats are commonly found in plants and fish. And they're usually oils because they're unsaturated. And then in general, plants have less use for fats slash oils than animals. So basically animals use fats and oils because it's more space efficient storage of energy, since as animals we have to kind of move around. But plants, they're just rooted to the ground in the same spot, and they can pretty much get as difficult to move as possible without there being consequences. So they're able to store a lot of their energy just in the form of starch. And yeah, saturated fats do tend to be solid at room temperature compared to unsaturated fats. So yeah, the answer is fats are composed of a glycerol linked to three fatty acids through glycosidic linkages. So all of this answer is correct except for glycosidic linkages. Those are used for carbohydrates instead. This is an ester bond. Next is which of the following about vitamins is not true? So it would be this. The last step of creating biologically active vitamin D in humans occurs in the liver. So the last step actually occurs in the kidney. Basically, kind of you have free kind of like inactive vitamin D floating around in your blood. And then, the first step of activating it occurs in the liver, and then the last step occurs in the kidney. So the rest of the answers are true. That like this first answer choice is true. So is the second one, and vitamin E is an antioxidant, and this is what antioxidants do. And B2 and B3 are involved with FADH2 and NADH, which are important electron carriers. So next, asking about surface area adaptations. So basically microvilli, they help increase surface area for absorbing nutrients from what you eat. Rugae in your stomach also increase surface area by quite a lot. And then plicae circularis, which are circular folds in the small intestine, kind of same story. And then, the shape of your alveoli basically has that same volume, but just increases the surface area by a lot to make gas exchange kind of more efficient. And then F. pili, however, their main function really is just to communicate between bacteria, share genetic information, stuff like that. So, the point of those is not to increase surface area. And then which of the following is false regarding eukaryotic cytoskeletons? So, this one is true. Cilia kind of do this kind of like rowing motion, like rowing a boat, and flagella kind of do like a fishtail motion that moves back and forth. So this one is incorrect. Cilia and flagella can have both 9 plus 2 and 9 plus 0 doublet structures. So cilia can have 9 plus 2 and 9 plus 0 doublet structures. Because, a non-motile cilia, like the primary cilia, it has a 9 plus 0 doublet structure, whereas flagella are only 9 plus 2 doublet structures. So, yeah. And then intermediate filaments do have the most permanent structure. Keratin is very, very strong. And then cortical microfilaments help maintain the cell shape and make up the cortex layer. Yeah, and those would be made out of actin. Fibronectin connects the extracellular matrix in the plasma membrane. That is true. So, now, which of the following correctly matches cell junctions with their function? So desmosomes don't use actin filaments, they use intermediate filaments. Gap junctions allow for these things, but not in plants. They're only used in animals. Tight junctions do bind cells together to make like a very tightly sealed layer, so that's true. Adherens junctions are not stronger than desmosomes, and plasmodesmata, their point is to basically serve as gap junctions for plants, kind of channels where things can flow through between cells. All right, so now let's look at this one. Steps for the activation of receptor tyrosine kinases. So we're going to start out with our individual monomers, which is step 4, and then we're going to have a signaling molecule bind, which is step 2. And then after that, we're going to have the monomers dimerize. Those two monomers are going to come together. And then essentially they're going to phosphorylate each other with ATP. And then finally, the receptor is considered activated, and then it causes a transduction cascade that leads to a cellular response. So next, which of the following is not true regarding apoptosis in the nematode C. elegans? So basically, this one is just knowing that apoptotic pathway. If you don't remember everything, go ahead and go back to the cell cycle lesson, take a look at it. But basically, this is true. And then CED9 does inhibit CED4. So yep. And the first molecule affected the first molecule affected is actually going to be CED4. Caspases, that's true. And then this is true as well. All right, so which of the following about the cell cycle is correct? Basically, this one is incorrect because your microtubules, in different organisms, some of them actually use kind of like a reeling mechanism, kind of like reeling in a fishing line. This one is correct. And the M phase actually makes up more close to like 10% of your normal cell cycle. A lot of that's the other time spent in G1, S, G2. Cyclin concentration is not going to peak during the S phase. It's going to peak, in mitosis. Cell plates in plants form from vesicles. That's not true. They form from vesicles from the Golgi. So, yeah. So, next up, which of the following correctly matches a photosynthesis mechanism with a plant that uses it? So, we're just going to look for one that's correct. Sugarcane does not use C3 photosynthesis. So sugarcane is technically a grass, and most of our grasses are going to use C4 photosynthesis. Millet does not use CAM photosynthesis. So CAM photosynthesis is used by kind of like your stereotypic like desert plants. Like pineapple uses CAM photosynthesis, so it doesn't use C4. Wheat doesn't use C4, it uses C3. and rice does indeed use C3 photosynthesis. So this is discussed in the photosynthesis lesson if you'd like to go back and review. Which of the following is true of cellular respiration in humans? So this is false. The first step is actually an intermediate step. This is true. It also has, a role in the citric acid cycle. This is false. A net of 2 ATPs are produced, but actually 4 ATPs are produced and 2 are spent. So, yep. And then ATP is produced during the formation of malate bifumarase. That's not true. Cytochrome C is found within the plasma membrane, whereas COQ is most commonly found outside the membrane. So that's actually reversed. Coenzyme Q is usually found within the membrane because it's hydrophobic, so it gets along well with the membrane, which is also like the inside of the membrane, which is also hydrophobic. Cytochrome C is water soluble. Okay, so now let's look at this one in a cross between these parents. What is the probability of producing an big A, small B, small b, small c offspring, given that the genes lie on separate chromosomes? So basically, this just tells us that there's no linkage, and we can just do some simple math to figure this out. So we can just look at the probability of getting, offspring with big A, and then with small b, small c, and we can multiply those together by the multiplication rule. So basically, the probability that you would get, a big A, small a offspring from, these parents would be 1/2. If you can just draw out the Punnett square for, the A gene. I could do some, kind of simple math, just saying that, we know 100% of the time we're going to get a big A from this parent, and half the time we're going to get a small a from this parent. So, 1/2 chance. And we're going to just do this process for the rest of them. So we get 1/2 times 1/2 times 1/4, and then that gives us 0.0625. Okay, so now we're going to look at a pedigree, and we're just going to see what the most likely mode of inheritance is. So, first thing we notice is, there are only males affected, so that kind of tips us off that it's probably something X-linked, because those tend to affect males more. Also, we do skip a generation. There's no one affected in the first generation, so it's probably recessive, and thus we are led to believe that it is X-linked recessive. Oops. So, moving on, which of the following incorrectly pairs an organism with its sex determination system? So let's just go through them. Grasshoppers do use the XO system. Bullet ants do use the haplodiploid system. Yellow jacket wasps are also part of Hymenoptera along with ants, and they're also going to use the haplodiploid system and not the XO system. Chickens do use the ZW system, and then baboons use the XY system. So the answer is C. All right. Which of the following about foundational DNA experiments is incorrect? So the answer is this one. So the Hershey and Chase actually did use P32 and S35, but not to label protein and DNA respectively. Those two are swapped. P32 is found in DNA as phosphorus. Is mostly found in DNA. And then S35 is used for proteins. So this one's just really knowing kind of what elements are found in what macromolecules. So, feel free to go back to the macromolecules video to review if you don't quite remember. And then all of the rest of them are correct. And these are experiments that are covered in the genetics lesson if you want to go look at those. So next is, which of the following statements about DNA damage and repair is correct? So it's much closer to 1 in 10 billion base pairs. Diamine dimers are formed from exposure to UV, not X-rays. This one's just not true. NER excises kind of a chunk, whereas mismatch repair is just kind of that one area. and this is pretty textbook, correct what it is, then that's not true. UV light does cause DNA damage. And then, which of the following is least likely to be involved in the packing of DNA? So, those first four, all used in the packing of DNA. These are all histones, so DNA is going to be wrapped around them. Condensing 1 and condensing 2, their job is basically to help with the looping of DNA involved in condensing into chromosomes. Okay, so which of the following statements about post-transcriptional modifications to mRNA is incorrect? So, just gonna go through them. So this is true, that's the definition of pre-mRNA. Spliceosomes don't contain microRNAs. They contain snRNAs. So that's what's wrong with this answer. Alternative splicing does allow for one gene to code for multiple products. And yeah, prokaryotes don't have introns. They don't have, any post-transcriptional modification mechanisms that deal with that. And then that is true about the 5' cap. And, okay, so for this one, just put things in order. So we start with the small ribosomal subunit binding first, and then the large ribosomal subunit binds next. And at this point, if you get those two, you already know what answer it is, but we can just keep on going. Basically, our codon in the A site is going to get recognized by a charged tRNA, and then from there, a peptide bond forms between that amino acid and the amino acid in the P site. So it's essentially like we're passing on that polypeptide we're building. And after that, ribosome shifts one codon down, tRNA leaves the E site, and then basically that just keeps on occurring until the ribosome reaches a stop codon, in which case a release factor is going to help free everything. Okay, which of the following statements about operons is correct? So basically all of these are incorrect. Binding of CRP makes it easier for RNA polymerase to bind. The lac operon is an inducible operon, and this is correct. The repressor of the lac operon binds to the promoter to inhibit transcription. It binds to the operator, not the promoter. And E. coli prefers glucose over lactose, and the lac operon is not constantly active. Okay, so we're just going to look at these diseases and see which ones are incorrectly matched. So Kuru is spread by prion. This is true. And sickle cell is autosomal recessive. Edwards syndrome is caused by non-disjunction, and it's trisomy 18. And yeah, the issue is PKU is not autosomal dominant, it's autosomal recessive. Next, we're just going to look at which of these is not a modified leaf. So onion bulbs are modified leaves. They're modified for storage, so lots of parenchyma. Sepals of a rose, stamens of a lily. So all of the whorls of a flower are modified leaves. So, these are cactus spines are also modified leaves. So grapevine tendrils, a little bit tricky, but basically the tendrils on a pea plant are modified leaves, but not the tendrils on a grapevine. Those are modified stems. So yeah, same thing goes for pumpkins, also modified stems. Okay, which of the following statements about plant tissue is correct? So basically, it's not the presence of parenchymal cells, it's the presence of sclerenchyma. Parenchyma cells are not, this is describing sclerenchyma and then offering ample support to fragile young shoots. So parenchyma does do that, but this description is of sclerenchyma. There are two types of parenchyma, scleroids and fibers. This is describing sclerenchyma, not parenchyma. Sclerenchyma is found in parts of a plant that are constantly growing. That's not true. Parenchyma are. So sclerenchyma, that would be pretty bad if they were found in parts of a plant that are constantly growing, because their main thing is, they're very stiff, and we don't want that in an area where you're constantly kind of growing and elongating and moving. Which of the following statements about plant vascular tissue is not correct? So this is correct. This is as well. So here it's not sieve cells that have them, it's sieve tube elements. And usually like for phloem and xylem, the names of cells that have elements in them, those are the ones that are mostly used by angiosperms, so like sieve tube elements, vessel elements. So yeah. Sieve cells are found in gymnosperms and are supported by albuminous cells. Cells of the phloem are alive. So, yep. Now we're just gonna see which ploidy is matched up wrong, and that would be C, the megasporangium is only 2N. Which of the following statements about the ABC hypothesis is not correct? So just draw out kind of a picture for the ABC hypothesis. If you don't know what it is, just look it up, maybe find a video on it. But, it's pretty easy to learn, and it makes lots of intuitive sense. So the whorl of sepals is not called the corolla, it's called the calyx. But it is controlled by the A gene. The corolla is a whorl of petals. Okay, so we're just looking for parts that are the apoplast. So cell walls, interior of xylem, extracellular spaces. Apoplast are basically things that are quote unquote dead. Plasmodesmata, cytoplasmic channels, those are very much like sort of alive in comparison, whereas cell walls, interior of xylem, extracellular spaces, those are kind of like empty and associated with like kind of dead cells or just like lack of cell. So yeah, that's what the apoplast would be. Now let's look at this one. So basically we're looking at what's unlikely to happen, so what's not true. So the answer is this one, because the opposite will happen. The stomata will actually close and try to save water, because that's one of the functions of ABA. So yeah, basically the rest of these are true. Auxin, cytokinin, the ratio of them determines whether or not your undifferentiated mass of tissue is going to grow roots or shoots. And if you have equal amounts, then you're going to get a callus, which is just kind of like a clump of undifferentiated cell. And then this is what gibberellins do. Ethylene, so this is kind of like the ethylene response that's being described right here. And then, IAA type of auxin is going to cause the shoot to do that. Basically, that's how, plants curving towards the light works. Okay, yeah, and then this one's just a pretty straight up just kind of look up a picture of, what the cross section of a stump or a plant would look like. In this case, it's a plant with secondary growth, so it would be something woody. You start with the pith in the middle, and then you basically just go primary xylem, secondary xylem, vascular cambium, and then primary phloem, secondary phloem, and then you get the periderm. So, yeah, or the court cambium and the periderm. Yeah. All right, which of the following statements regarding capillaries is not correct? So all of these are going to be true. This is described in the cardiovascular or the circulatory system video. And then this one is not true. Parasites, they just kind of occur occasionally on the capillaries, definitely not a full layer. So endothelial cells, which like basically form up the capillaries, those are, I would say, like kind of in a more complete layer, as they literally make up what a capillary is, compared to parasites. And then, okay, so when we look at this one, bile helps with the digestion of fat. So we're looking for a food that, where it's really fat heavy, and if Mr. Collins eats that, then he wouldn't be able to really digest that properly. So, guacamole from avocados, very fat heavy. So, basically, if he has a bile blockage that doesn't allow like bile to come out and do its thing, he's gonna have issue digesting fat. So he does not want to be eating guacamole. which of the following statements about the adaptive immune system is incorrect? So basically the answer is this. The proteins are just swapped. Perforin breaks down the membranes, granzymes initiate apoptosis. Everything else is correct. Which of these organisms is most likely to use the following organ? So this is a flame bulb, from the excretory system. It's mostly used by flatworms, so that's what we're going to look for. Dugesia japonica, which is a planarian, kind of flatworm. Those are going to use flame bulbs or, Protonephridia. Okay, so this one is just about kind of the endocrine system in moths and basically how that controls their molting. So basically, we're just gonna look at all of these, and for one that is true. So most of these are not very logical. If you know what's kind of going on with the system here, just look up kind of a diagram of all of these interactions, and all of this will make sense. But basically, this is what happens. It's juvenile hormone being produced at high levels that's keeping that kind of metamorphosis from being complete. And then for this one, again, just look up a picture of an action potential. It should help a lot. So basically, we're above the threshold potential after, we reach that threshold and all of the sodium channels open. So we're going to be above it when there's a massive Na plus influx, which is 3, and we're also going to be above it when we're going to start getting Na plus channels getting deactivated, and then K plus channels start opening. So that is number 4. And this one, basically just know your embryonic brain regions and kind of what they lead to. So the telencephalon does not lead to the pons. The diencephalon doesn't lead to the cerebral cortex. The telencephalon does. Metencephalon doesn't lead to the medulla oblongata. The myelencephalon does. Mesencephalon actually does lead to the midbrain. And the myelencephalon doesn't lead to the cerebellum. So the answer is D. And then for this one, basically you can kind of search up a picture of this. But basically, depending on where you cut or sever kind of these pathways, you're going to have different effects on the vision. So if we sever at the optic chiasm, we're going to lose sight from kind of like the leftmost and the rightmost, and the person will still be able to see kind of the inner region, of their visual field. And then this one's just kind of knowing your glial cells, and the issue with E here is that it's not located in the PNS, it's located in the CNS. And then this one is knowing kind of what a sarcomere looks like. So basically, there's an acronym for it, MHAIZ, maze. You can look up a picture of it. But basically, the A band doesn't contain only myosin, it contains all the myosin. Whereas the H zone is the one that contains only myosin. This one, just basically slow twitch muscle fibers have less sarcoplasmic reticulum. That's why they twitch so slowly, right? Because they're able to hold kind of that longer, more sustained, contraction. And they have higher myoglobin content because they have more of a need for oxygen. Since slow twitch muscle fibers are all oxidative, so they all use cellular respiration, compared to these glycolytic muscle fibers that don't depend on oxygen content. So we're going to have a lot more myoglobin when we need to use oxygen, right? So this one, this is social learning. You're learning from kind of the elders in the group. That's what social learning is. And for this one, the answer is polyandry. Polyandry is when there's one female, multiple males. Polygyny is the opposite. So basically, in the polyandry system, the females are going to be more ornamented, And, in polygyny, it's reversed. Which of the following statements about dispersion patterns is not correct? So all of these are very correct. When organisms are clumping around a food source, that's going to be a clumped pattern of dispersion. Highly territorial animals like penguins are likely to have uniform distributions because they're going to keep their own territory kind of around them in like a similar sized area. And if you just put them all in one spot, they're going to quickly kind of fight out their own territory and they're going to be uniformly spaced. As a result, plants with wind-blown seeds are likely to have random distributions. Yeah, so this just depends on where the wind blows their seeds, right? And that's going to be random. Pack animals have clump distributions, yeah, because they want to be with their pack, they want to hunt with their pack, and as a result, they're going to be in groups as opposed to like uniformed or some other distribution. Allelopathic plants like black walnut typically have random distributions, so that's not true. Allelopathy is basically when these plants, they secrete something that makes it hard for other plants to grow in the area. And that's going to result in more of a uniform distribution. Like no other black walnut trees are going to be able to grow that immediate circle around a black walnut tree where it's preventing other plants from growing there. And then this one's just knowing your survivorship curves. So the one that is incorrectly matched is mussels. They are type 3. Just look up, a survivorship curve and then know some of the examples, kind of why they're that way, and this question will make lots of sense. This one, an R-selected species, kind of the gist there is make a lot of, babies, and then those babies, most of them won't survive, but because you made a lot, then, you'll end up with a decent number. So boom bust cycles, basically there's a lot and it suddenly falls. So that is associated with our selected species. Shorter life expectancy, that's true. The reason why there's so much reproduction is because it's not expected for these organisms to live very long, and thus you need more to kind of counteract that. Lives in an unstable environment, so that's true. Our selected species usually do. And then relatively large size produce few offspring. Those are characteristics of K-selected, not R-selected. Okay, so for this one, we're just looking at what type of mimicry is being described, and it's Baitian. Basically, a harmless animal. It mimics a harmful animal to make itself look less desirable as food. And that is an example of Baitian mimicry. Mullerian is, when, like poisonous animals look like each other, and that helps kind of reinforce that idea. Emslian is when a more poisonous animal tries to look like a less poisonous one. Aggressive has to do with predation. Cryptic coloration is just camouflage. So if you read this scenario, you're going to know that this is commensalism. Basically, the flower shop doesn't benefit from this transaction, but Charlotte does, and you can look up some really neat charts online where it shows basically like who benefits from each of these relations. In commensalism, it's going to be like a 0 and a plus because one person or one organism doesn't benefit, but it doesn't get harmed, and then the other one does. So that's commensalism. And then, which of the following statements about plant phylogeny is not correct? So all of these are correct, but monocots are not considered basal angiosperms. There are actually species that are, like Anis is kind of in there, but monocots are not. And then lastly, which of the following characteristics are typical of monocots? 2 cotyledons, that's dicots, that's what they're named after. Parallel leaves, yeah, that's typical of monocots. Taproot, dicot, ring-like vascular tissue, that's dicot. Monocots have like a scattered pattern. And then even number of flower petals, that's something you're only going to find in dicots. So yeah, that concludes the test. I really hope this was helpful, and good luck on your USABO journeys. Good luck if you're taking opens soon. I hope that you're able to, kind of get some studying in these last few days and you're able to do your best on the test. I'll be posting more, so, if this helped you, please leave a comment or a like, and, please subscribe for more content. Thank you.